So I was going to talk about some of the imaging research that's been done in blepharospasm and that's being done, some of which here at University of Colorado. It is unlikely that I'm going to clear all things up that Dr. Hallett mentioned, and might even highlight that there is this confusion, and but that just leaves the door open for more research to be done. So we all want to know what causes blepharospasm, and the better we know what causes it, the better we can develop treatments or a cure. And you heard from Dr. Fawn earlier about genes and environment, and Dr. Hallett, and how these come together to create a disease, something like blepharospasm. And these factors, genes, and environment, we know have some effect on the brain. And so we try to look at the brain in different ways. As we mentioned, there's pathology studies that we don't have enough brains to really, or we're not looking at them in the right way, so we still need more. So please do donate when you're done and using them. And then the other way to do it, but while you're alive, is to use neuroimaging. So neuroimaging is when we use a variety of these different types of tools. And so you may have heard some of these. So CT or CAT scanner that uses radiation, MRI scanner that essentially turns you into a magnetized antenna, the PET CT scanners that use some radiation to help image things, and EEG and MEG. These are other ways to look at brain activity to tell us what the brain is doing. And they can tell us both the structure of the brain and also some of the function of the brain. And so what I was going to talk about is how the imaging tools that we have are being used to examine those potential causes of dystonia, those substrates. And so Dr. Hallett mentioned we think that there's reduced inhibition that plays a role, that increased neuroplasticity might contribute to the disorder, and that the sensory and motor integration seems to be abnormal. And there are a number of disorders that have also reduced inhibition or abnormal neuroplasticity, and so we wonder also if it's sort of the confluence of these factors that creates dystonia, or if just dystonia gives us signs that these three pathophysiological themes are present. So I'm going to first talk about reduced inhibition, and so how can we use imaging to study that in the brains of those with dystonia? And so we can use positron emission tomography, or PET, and also magnetic resonance spectroscopy, which is a fancy MRI technique to look into inhibition of the brain. And so what you heard earlier, that there's potentially a hyper-excitable eyelid closure, that it's excitable because there's not enough inhibition of the reflex in the eye. And so that might underlie blepharospasm. And this is consistent with other dystonias that have been associated with deficient inhibitory function in the brain. And so inhibition in the brain is really one of the main ways it's controlled is with what's called GABA. And GABA is a brain chemical in the brain that's like the red light, the stop signal. And so it helps inhibit brain activity, while the green light, is glutamate, and that's a brain chemical or neurotransmitter that excites the brain. And brain cells communicate with each other with chemicals, and so the GABA chemical is released and has what we call receptors on the other side, on the neighboring brain cell that it's communicating with. And so one way to investigate the GABA signaling is to look at the receptors for this inhibitory chemical. And we can use positron emission tomography to do that. And that looks something like this, a very colorful rainbow image of all the GABA receptors in the brain. And as you can see, there's a lot of them, and they're spread out throughout. So what we do is we compare a group of what we call healthy controls, those who don't have dystonia and those who do have dystonia. And there's been at least one report of this by a group. And they looked at DYT1 dystonia patients and some sporadic dystonia patients. Unfortunately, none of these there was only 5 in this group, and none of them had blepharospasm. And they're also kind of younger, so it's not sure how much this study is indicative of blepharospasm. But they did at least note that decreased GABA receptor binding was seen in some of the motor areas and some other areas like the what we call the anterior cingulate area. This is other parts of the brain that are involved in both sensory and motor functions. So I wanted to extend this study, and so here we've been doing a very similar type of technique, looking at GABA binding in the brains of patients who have cervical dystonia. So this is not blepharospasm, this is cervical dystonia, but as we mentioned, sometimes there's an overlap between these conditions and they can spread. And when you look at the whole brain at that rainbow image that I showed you, the whole brain does show decreased GABA binding, so diminished inhibition in those with dystonia. And if you look at the specific areas that have the most decrease in that GABA binding, you can see this is called a premotor area in the brain. This is near what we call the insula cortex, which plays a role in integrating sensory stimulus, like it's cold in this room or it's windy outside, and then causes actions to happen from that. And the cerebellum, which has also been indicated in dystonia. And picking just one type of dystonia helps with comparing how the GABA system is related to motor severity, and looking at what we call a Twister score, which is the severity of cervical dystonia in these patients. And this is 10 patients so far that were studied here, and a very clear decrease in GABA binding or GABA signaling when with more severe symptoms, telling us that there seems to be some link. May not be causative, meaning that this might not be the cause, but we at least see that it's tightly linked to severity of motor symptoms. So some work in the future will be to finish that study, which we are only 2 scans away from finishing, and then hopefully those results will be out soon. But another thing to look at is that back to the GABA signaling, when there's these receptors, there's also the GABA itself. And so how much GABA there is may change how much GABA binds to these receptors. And so we can look and use tools to just tell us how much GABA is in this region. And that's just done with a standard MRI scanner, but using a fancy type of scan with it. And in this case, I would like to use my patients who show decreased binding in these areas and find out, is there also decreased GABA in those areas? And that'll give us more information about the inhibition system in the brain. So the other substrate that was mentioned is increased plasticity, and we can presumably use structural MRI techniques to kind of look at whether the changes in brain connections and size alter the structure of the brain. And so, plasticity, what we mean by that, it's a term that's talking about the ability of brain cells to change, and it's this change in their size or their numbers of connections or how they're organized. And it's thought that blepharospasm might be caused by changes in how these neurons connect to each other, we think an increased neuroplasticity. And so does that manifest as changes in brain volumes? And we can use an MRI scanner to examine this and see if that holds true. Unfortunately, as Dr. Haller mentioned earlier, there are a variety of studies, and these are from his slides, so I didn't have to go cut out the images somewhere else, but they showed when we first looked at it, it looked just like we might think, that the putamen was enlarged, and that fits nicely. But we know in science and in research, if you check it a second time, then you get a different answer. And so they got the opposite type of answer in the other direction. And then there's also seems to be cortical changes. And there's been a variety of studies that keep finding different answers. And it just shows you the complexities of research, that we have to have a particular type of patient. We have to have enough patients and use methods that we can all agree upon. And so that might play some role in why we see a difference between these different studies. It could be that there is no standard area that can be found that we do see changes, but we might see different areas of changes in different groups of patients. And one theory to put out there is that maybe the sizes of our brain areas don't change, but maybe the shapes do. And so perhaps future studies that look at actually the shape of a structure. So these are some of what we're talking about, the basal ganglia, and this is the yellow is the putamen. So you can imagine that if there's maybe some increase in plasticity here, but decreased here, that the overall volume wouldn't change. And so it's actually no change. But if you examine the shape of it, and there are fancy analytical methods used now to look at shapes and then compare them between groups of subjects. So we may be able to tell in the future that actually the volumes aren't really different, but how they're forming and what shape they are is different. So the third type of abnormality that seems to be present in dystonia and blepharospasm is abnormal sensory motor integration. And when you're looking at sensory motor function together over time, one method to use is called functional MRI. Functional MRI is looks at oxygen changes in the brain and assumes that if there's oxygen changes, that's probably brain activity changes, and we can get a sense about how a brain is functioning under certain circumstances. The meaning of the sensory motor integration, just to help define that, is the network that's involved in integrating body sensations with motor responses. And there's evidence across a variety of dystonias, blepharospasm as well as others, that this integration is abnormal, it's altered in dystonia. And so one way to test this is what the BEBRF helped fund. It was a study that I did where we're looking at trying to actually look at the brain's response to a sensory stimulus. And so in this case, we use a air puff, and we have them in the scanner with a lot of equipment, including a camera that captures this, and EMG leads that looks at the motor activity. And then you can see this right here, the puff. So they just got a puff, and they start to blink, and then they're trying to keep their eye open, and there's some spasms going on, and then they just had a puff and a longer, prolonged spasm. And so this is a way to try to see if we can investigate what is going on in the brain when they're responding to a sensory stimulus. So completed the study, and we're in the process of completing some analysis, and there was 15 healthy controls. And if you look at the activity of the brain, this is actually some sensory areas. This is the insula and the cerebellum. And so these are areas that you might think would be involved in getting puffed in the eye with air. So there's a sensory cortex response, and there's actually some putamen or motor response here as well, which may reflect the actual motor signal to close the eye. When you look at the 15 blepharospasm patients, they have the same pattern. So that was, their brains are responding in a similar fashion. Of course, it triggers spasms and other problems. So if we compare the controls to the patients, we see a couple areas, at least in this comparison, that do look different, and that they're not in areas that have been thought much in the past, but there is in the occipital, sort of visual cortex region, there's increased activity. There's also some in some of the frontal regions, and this one might actually be a motor region in the eye, and so or the eye region of the brain that is associated with those increased responses, the increased motor responses. So this still needs some interpretation, and this is pretty new analysis, so that'll be interesting to see if it holds when we apply more strict standards to our findings. So the other thing to do, since we have them in the scanner and we're checking their EMG signal as they're lying there, is just say, well, if we just don't puff them with eye and the eye is just spasming away, what is the brain doing when they're having these spasms? And so we can take this signal from these electrodes that measure the muscle activity and match them with the brain activity and try to see, what is the brain network or circuit that's being active at that time. So we've done this with 15 blepharospasm patients, and it came out somewhat what might be expected. So there's cerebellum, there's some what we call post-central or sensory cortex here, and what's called the supplementary motor cortex here. So this shows that there's this sort of important circuit that seems to be involved with driving the blinking and spasms in these patients. And if you consider this sort of a network, then the strength of that network decreases with increasing motor severity. And I did this with each and every area and each and every scale that I used. And so this, as we were saying, the abnormal integration, so the more the circuit is not together, then there's more severe symptoms. So I think this kind of provides some direct evidence that there is network dysfunction between sensory areas and motor areas. So in summary, it seems that some of the imaging research is increasing our understanding. Those three pathophysiological themes that we talked about that have been described in dystonia, the reduced inhibition, increased neuroplasticity, and abnormal sensory motor integration. But still, as we've all been saying, there needs to be more research because there's still many challenges and questions that we need to answer. And if you are interested in participating, this would be fantastic. We are enrolling for the Dystonia Coalition Project 4 that you heard a little bit about. That's called Diagnostic and Rating Tools for Blepharospasm. And this study just, if you are a spouse or partner of someone who has blepharospasm, there's important needs for those healthy controls, those who don't have them. So you can also do something to help contribute to advance the research, even if you don't have blepharospasm. And we're also enrolling for what's called a natural history and biorepository study of focal dystonia, including blepharospasm. And that's to help us both better understand how does it spread. Does it spread differently in people with blepharospasm and other conditions? And also to look at the genetics that we also heard about that we need to do more research in. And Erica Shelton is my research assistant. She's out there. She has a booth, and you can contact her here at this number or email, or see her today if you're interested in participating. And I just want to make sure that I thank the BEBRF who helped fund the fMRI studies the functional MRI studies that I presented here briefly, and also the Dystonia Coalition Dana Foundation and the Dystonia Medical Research Foundation, which have played a part in helping funding some of this research that I've done. All right, well, that's it, so thank you.